We're really excited about this. This is the first time we've done an organic research updates kind of lightning talk or flash talk series. So we'll have four presenters today share updates on their organic research in California. We'll have about 5 to 7 minutes per person with an opportunity to ask probably one or two questions depending on the length. So with that, we'll jump in and I'll have Justin share his screen. Justin Valliere is an assistant professor of cooperative extension and invasive weed and restoration ecology at UC Davis. His research and extension program seeks to develop science-based solutions to invasive species and restoration challenges through close collaboration with practitioners across the state. And I'll hand it over to you.
Great. Thanks, Krista. Can you all see my slides in the full view? Great. Okay. Yeah, so as Krista said, I am primarily a restoration ecologist, but so I work mostly in wildlands, but I'm really interested in how we can use native plants in agricultural settings to support growers, but also to support other, biodiversity conservation sustainability goals. So I think, as everyone here can appreciate, field edges are a ubiquitous feature of landscapes in the Central Valley, but as well as other agricultural landscapes. And so these are really important, right, to serve as like field boundaries, to get equipment and water and resources in and out of fields. But this is also problematic because they can lead to, dust, erosion, and as relevant to this talk, they can also be important conduits for weeds and harbor a lot of weeds that then end up in our fields and orchards. And so this is also problematic too, because if you think about areas like the Central Valley, some of the only habitat that we have for native species is these field edges, but they're often filled with non-native noxious weeds. And so I'm sure many people here can appreciate the value of native hedgerows. This might not be new information for a lot of folks, but native hedgerows aren't super widely adopted throughout the state. I think that is changing, and some of this data might be a little bit outdated right now. But if we look at the practices that most growers use to control weeds on those field edges, we see things like mowing. So the blue here is current methods, the yellow is past methods, and then the gray bars is never. So these are percentages. So we can see a high percentage of growers surveyed in the Central Valley used mowing and herbicides or disking, to a lesser extent burning. But then native hedgerows or planting other types of native plant species as competitive plantings are really underutilized. And so this is work that Rachel Long, who is the co-author on this project with me. She's an emeritus farm advisor. This is some of her previous research aimed at understanding, well, why aren't native hedgerows more widely adopted? And so when she surveyed growers and asked about what things they were most concerned about, you can see that weeds was the top concern among growers. And so Rachel designed this project, which I later joined, to understand, do native hedgerows actually lead to greater weed abundance? Or conversely, can native hedgerows actually be used as a proactive measure to prevent weed intrusion into fields? And so Rachel designed this really great study and collected all the data where she identified 20 farms, and that included orchards as well as row crops. And each of these fields had a native hedgerow on one edge of the field and then conventional management on the opposite end of the field. So kind of this paired design. And sampled these fields or these farms from 2015 to 2017. This included winter and summer sampling to capture species that are active at different times of the year in these systems. And so all of these sites were located in the Sacramento Valley. And this is a diagram explaining how we did our sampling. So as I mentioned, there was a native hedgerow on one side of the field, conventional field border on the other. And then we ran transects at different zones within the field. So right on the field edge, and then 10 meters in, 75 meters in, and so on. For row crops, it was just simply intervals into the field. For the orchards, we did in the avenues in between trees as well as underneath the trees as well. And so I'm not going to present all the data from each one of those zones within the fields. This is just focused on the percent reduction in weed cover richness, so the number of unique weed species and weed frequency just at the directly at the field edge. So you can see for both field crops and for orchards, native hedgerows reduced weeds in terms of their abundance, in terms of the number of species, and the frequency or the density of weeds. So over 50% reduction compared to that conventionally managed edge. And for row crops, this benefit extended slightly into the field. It wasn't a dramatic response, and that's because folks use intensive weed management within the fields themselves. But within orchards, it was really interesting. That benefit extended sometimes like 75 meters into the orchard. So it is true, in fact, that like these hedgerows aren't harboring weeds or increasing weed intrusion into fields. They're actually doing the opposite. Um, we also looked at the percent cover of native plant species versus weed cover. So native cover in the hedgerow is shown on the x-axis, and then weed cover on the y-axis. And not surprisingly, what you can see is like the greater canopy cover you have in your native hedgerows, the lower weed abundance. So getting a high enough density of plants established and mature is really key for having this weed suppressive effect early on. When you have young plants, you're going to have to undertake some weed control efforts as those mature woody plant species or perennial species get established. And then importantly too, there's also an economic benefit. So certainly you'll have to pay costs up front to establish these native hedgerows. There are incentive programs like the Healthy Soils program to help cover some of these costs. And that'll be the cost of the plants, the labor. Irrigation is probably really important early on as well. But once these are established, you can see that the management costs for hedgerows, this is self-reported data from the growers that were included in this study, a really dramatic cost in weed management efforts compared to conventionally managed fields, which have to just constantly be retreated every season or multiple times per season. And then when we look at herbicide use, this is another really promising result and very relevant to this meeting here. We also asked what types of practices growers that were using hedgerows versus conventional management, how often they were using herbicides. And you can see for conventionally managed field edges, a little more than half, or a little less than half, were using herbicides, but none of the growers that were using native hedgerows reported having to use herbicides in those hedgerows themselves. That didn't mean that they weren't using herbicides elsewhere on their field. Not all growers were strictly organic, but I think this is a really promising result. So collectively, hopefully this relieves some of that concern about weeds being an issue in native hedgerows and will lead to increased adoption over time. So hopefully I've convinced you that native hedgerows are indeed an effective weed management strategy. So once established, they reduce the cover, the diversity, and the density of weeds in field borders. The greater the density of your native plants, the greater the benefit. And as I mentioned, for orchards, this can extend far into the fields. And then native hedgerows can also reduce weed management costs and labor and herbicide reliance on herbicide. And this is in addition to all those other great ecological benefits that we know hedgerows can serve. So in terms of enhancing biodiversity or expanding wildlife habitat in agricultural settings, improving erosion control and soil carbon sequestration, as well as supporting beneficial insects. So natural enemies that are really important for pest control, as well as pollinators that are really important for agricultural production. In addition to those benefits for weed management, as I mentioned. And with that, I did forget to time myself, so hopefully that was okay. And I'm happy to take any questions.
Thanks so much. That was awesome. Really exciting to see some of the research showing all the multiple benefits and potential agronomic roles that hedgerows can play. I think we have time for maybe one quick question. See, Katharina had posted. Did you want to ask a question?
Sure. I had two. you could choose either one of these, Justin, but one
Sure.
is hedgerows. I know sometimes are planted with an understory of native grasses, and that might also outcompete weeds. So I'm wondering if your hedgerows had native grasses or not. And, we were excited to have you talk because even though it's a conventional system, hedgerows are important elements of organic systems. And so do you see this study playing out differently in on an organic annual or perennial farm?
I think I can briefly answer both of those. So I think yes, certainly you can see benefits on organic farms because it's still going to reduce costs, and the need for using equipment in these hedgerows to if you're using disking or other non-chemical methods, mowing. And then in terms of the native perennial grasses, I don't know for sure for all of these hedgerows. Some I suspect did have herbaceous plants, and I think especially early on, having a combination of woody plants and herbaceous perennials could be really important. Um, and I do know that the Rachel and John Anderson created a document years ago on providing advice and guidance on how to develop these hedgerows, and they do suggest including perennial grasses intermixed with those woody plant species.
Very cool. Thank you so much.
Yeah, of course. I am
Yeah,
just going to make one shameless plug for the new ecological restoration rick that Leslie Roche and I just launched. So I'm going to drop the link in the chat there, and if there's a page on hedgerows in there, but a lot of other information about ecological restoration if folks are interested.
thank you so much. Thanks for taking the time to share your research updates with
Yeah,
us.
thanks so much for having me.
Yeah. All right. So we're gonna move. Oh, I see, Ben. If you have a question, if you could post it in the chat, and we'll actually collect additional questions and share those out with presenters, just so for the sake of trying to keep on. Um, so next up, we're going to have Darryl Wong present. He is the executive director of the Center for Agroecology at UC Santa Cruz, where he leads the center's research, education, and extension programs. I will keep it at that, and I'll have Darryl share his screen and feel free to take off.
All right, can folks see my screen? We're all set. Okay, look, I'm starting my own timer, Krista. Let's see, I actually managed to do it. Well, I'll say I maybe feel a little bit like the odd one out in this bunch. My role as the executive director at the UC Santa Cruz Center for Agroecology is really, I think, to support a lot of the scientific efforts and research that's happening here. I don't do as much of the actual data collection and analysis, so my presentation is going to be a little bit like research light. I'm going to talk about the stuff that we're doing and we've been working on. We've started a number of projects in the last couple of years. So unfortunately, I won't have, I think, great slides and data to show as maybe some of our other more professional scientific presenters will, but I'll give it a shot. Um, let's see. Okay, so just as a quick reminder for folks that don't know, right, we have been around for almost 60 years, but just formally joined the Ag Experiment Station Network in 2022 as a campus at UC Santa Cruz. And there's a lot of, I think, really exciting work that's been slowly building as part of that to engage both with cooperative extension, but also the research enterprise. So a couple of things that I just wanted to, I'm going to highlight a couple of projects that the center itself is maybe particularly engaged in, and then as well point to a couple of research, kind of loose research projects that some affiliated faculty are working on, kind of as more of like a overview of things that are happening. Again, without, unfortunately, I can't get too deep into maybe the specifics or at least the results of some of these trials. So many folks will be familiar with the this plot over here is work that was done at Santa Cruz for many years on anaerobic soil disinfestation to really support organic production of strawberries, to support soil-borne diseases. Currently, we have a project through the CDFA that Dr. Jenny Broom, who's an affiliated faculty with us, is now running that's really working on steam treatment for strawberries. So they have built this kind of steam rig to try to manage some of the soil-borne diseases that aren't as easily managed with anaerobic soil disinfestation with this kind of high steam treatment. So that's something we're just in year one of that. I think we're just starting to take some of the first trial plots were put in the fall and starting to take some data on that, but just continued work in the kind of strawberry soil-borne disease pathway. And some of this stuff I just put in, right? There's a lot of conversation in our area in particular about how do we keep kind of chemicals, pesticides, and herbicides away, especially from schools around our region. And so we're really excited that some of this work is supporting that. Um, Folks that have maybe heard me present previously will know, a lot of my work, specifically for my dissertation, this was now about 5 years ago, was really on organic no-till for specialty crop systems. So we ran a 3-year trial looking at specialty crops in a no-till system that had a very low residue cover. We used white mustard as our no-till cover that was roller crimped. And just a couple pictures here of kind of the difference in growth. Even though we didn't have really strong weed competition across our no-till plots, in our shallow till and full till, you can see there was just so much more kind of nutrient availability. We're still trying to tease out what some of those issues were. And even in the subsequent buckwheat cover crop, we saw this same kind of nutrient signature. So I do have data from this. I don't have it ready to present for you all, but the bottom line is that across both lettuce and broccoli crops, we basically saw like total crop failures in this organic system, despite doing 2 and at times 3 times our normal application of pelletized fertilizer into these systems. We still couldn't see any commensurate yield response. Um, and that was over kind of a 3-year process. And so my takeaway from this, right, when people continue to say, right, we want to do no-till on organic systems, we think it's good. My sense is like it's just not there for prime time yet. We learned a lot from how much this system failed, and we're still trying some new things. So the update on that is that one thing we've continued to try is, and something that I just like to reiterate for anybody that's actually thinking about doing this stuff is, you know, for folks that have used a new piece of equipment or tried something, like it just takes some time to understand how this works. And I'll say, right, for a number of years, we've been trying to work with this no-till transplanter that comes out of the Midwest from Mechanical Transplant Company. And I really feel like this past year was one of the first years we actually got the sequence and the deep moisture right to actually pull it through a field that had a rolled Merced rye cover crop and actually establish a stand. That said, right, those of you that will see what this stand, it looks pretty when you plant, but we had tremendous issues with weed pressure and regrowth. This was in a field that we just tried just Merced rye in a single year, so this wasn't a field that had been no-till year over year. But again, we're continuing to play with the implements to try to understand, I think, how quickly they work. All right, I got to move faster. The big takeaway for the no-till research now is we've moved into looking specifically how cover crops operate in these systems. And in particular, this here is a section of a research plot, 1 rep, that is all no-till, but this section back here has no compost applied, and this section here has 100 tons per acre applied. What that ended up equating to is that in our no-till plots where we applied that compost, we saw similar biomass growth as we did with kind of full tillage or tillage without compost. So it actually took away the tillage effect. And that's really encouraging for us to see some type of response in these no-till systems, even across cover crops. I wanted to point here, some folks have seen this slide, but some of the folks that we have working at Santa Cruz are doing things like PET scans. These are scans that typically are used in medical situations, but they've developed instrumentation to actually look at soils in situ. So you can actually see, right, what the aggregation is around, this is a sample of no-till soil from this plot, and this is a sample of the tilled soil. So really beginning to kind of quantify what's that aggregation structure with some kind of novel techniques. Let me move just a little bit more quickly. We're doing some work on cover cropping. So this is the cover crop replicated trial where we're looking at different cover crop plantings. Is there a trade-off between planting early and applying irrigation with the cost of that, but what it gives you in terms of nitrogen scavenging? Again, thinking about a later established cover crop and some of the leaching that might happen before that has an opportunity to suck up some of the nitrogen available in the warm soils. This is that same field, but paired with like a winter squash that we grow through the season. But this is work that Hannah Waterhouse is doing. Some of the paired work that's not quite formal is that we're beginning to use some drones to combine both the below ground and biomass sampling across these treatments with above ground modeling from these drones so that if they're able to take the right spectral imaging and get the right data, we can begin to create models where you might be able to get a good estimation of what your biomass and nitrogen kind of scavenging potential is just based on a drone flight versus all of these kind of more intense measurements from all these data loggers in the field. The last I'll show, we are continuing to work on small farm robotics. We have a 4-year grant with the California Department of Food and Ag that we're working on right now. We have a cohort of 6 growers that we're working to actually design transplanters, weeders, and harvest trailers for these kind of tractor replacement platforms. And that's very much in progress right now. We just started that this fall. So yeah, if folks are interested, right, we have lots of other research projects that are happening on our website. Um, and I'll go ahead and skip that. I'll skip this. These are some other things that other faculty are working on, but stuff that I think what I just wanted to point to here, right? Some folks with kind of medical background are working on biomimetic leaf wetness sensors. So you can create a leaf wetness sensor that will actually mimic the plant leaf rather than something that's a little bit more, right? Not as particular to like how a kale leaf would shed water versus a lettuce leaf. And that could be something you could put specifically in a greenhouse for in organic systems to make sure you stay ahead of those disease curves. And some things around, yeah, some other kind of intensive uh, Internet of Things. The last thing I'll say, and this is a little bit of like my own shameless pitch, is just to say the campus at this point, I'm excited to say, is we're actively pursuing expansion onto over 200 acres of agricultural land, which is right adjacent to our coastal campus. So this is like a mile from our campus. So in terms of the research talk and working with technical assistance providers in the state, I am hoping, we've made incredible progress on the fundraising for this amazing piece of property. And I hope that in the future, this will be an opportunity for those of you that are looking for partnerships with a UC campus and the researchers that will have, I think, a lot more space than we've had previously. So I will stop there.
Awesome. Thanks so much. It got my head. My brain is spinning. There's so many cool things happening at UCSC Center for Agroecology. So thanks for taking the time, Darryl, to give us an overview of all of the different activities happening. I think we have quick time for one question. I saw Lauren in the chat. Darryl, who at UCSC is running those soil scans to profile aggregation?
It was Wei Xin Cheng, but he just retired. He's still going to be doing some. And I will find the name of the other faculty that's working on that and I'll put it in the chat. I cannot remember her name right now.
Awesome. Thanks so much. Yeah, I think that big round of applause for Darryl. Thanks so much. We're gonna move on, stay on time, and now we're gonna hear from Charlie Brummer. Charlie is a professor at UC Davis and director of the Center for Plant Breeding. He also supports the Student Collaborative Plant Breeding Education or SCOPE project at UC Davis, and I'll hand it over to him to share a little bit more about what's been going on.
All right. Is that coming through okay?
Yep, that looks good to me.
Oh. Can you hear me?
Yep, I can hear you.
Oh, okay. It said it was
Yep, good to
muted,
go.
but I didn't think so. Okay. All right. Well, no, I'm actually in the middle of my talk here. This isn't good. Oh, I can't even. What's going on here? Okay. Somehow that started in the middle. Let me start from the beginning. All right. So I'm a plant breeder at UC Davis. I work on a number of species, some of them under organic conditions and some not. But they are alfalfa, hemp, spinach, shown there in the greenhouse in some crossing tents, and zinnias and some other crops related with scope, which I'll talk about in a minute. My program, it covers the state from the Oregon border down to the Mexican border with all kinds of cool stuff going on with on-farm alfalfa trials, breeding in alfalfa for horses with this rancher up in Herlong. I do way up north here, some grass variety trials on ranches. We're doing some on-farm cadmium uptake studies in spinach and alfalfa breeding down here in the Imperial Valley as an example of some of the things that we have going on. I'm really into alfalfa. If any of you know me, you know that I'm really into alfalfa. And part of the reason I'm into it is because it's beautiful. And here are some alfalfa germplasm pictures. This is one of my graduate students, Charles, here in the middle, looking at what we think might be a very nice ornamental yellow flowered alfalfa. But you look at these different flower colors, God, it's just the most beautiful crop. And so we're doing a lot of different work on alfalfa germplasm, trying to develop new varieties, largely for hay production under irrigated conditions, but also for other things as well. This in the middle is a creeping rooted alfalfa, an unusual phenotype, something that we're trying to introduce into more high yielding alfalfa, something that may be giving more persistence to alfalfa. And it certainly is something in rangelands that can be quite useful. So we're doing quite a bit of work on rangeland alfalfa for non-irrigated conditions, as well as on the left here, one of my former students, now an alfalfa breeder in Australia, Matt Francis, selecting alfalfa under both conventional and organic conditions for high yield, high production. So now I want to switch though and talk about Scope, which is exclusively organic. This is the Student Collaborative Organic Plant Breeding Education Program that's been funded several rounds by the USDA NIFA Organic Research and Extension Program. And hopefully we're going to get more funding this year. We'll see what USDA has to say about that. This is a collaboration with the student farm here at UC Davis, as well as UC Santa Cruz and CalPoly Pomona. And the idea with Scope is to educate students, these are both undergraduates and graduate students, to get hands-on plant breeding experience. These are really student-run programs. So the students come up with breeding goals, make crosses, put out field plots, and collect data and so on, with some advice and help from faculty and especially staff. We have some incredible staff here at UC Davis who have kind of maintained this program over several years. But I've always been interested in making sure that even though we're doing this field work that's helping students understand the breeding process, that we end up with varieties that come out at the end. And so we've been releasing varieties bred in organic conditions for organic systems. And these varieties have no IP restrictions that prevent further breeding. And some of them are fully public releases that anyone can do whatever they want to with them. Let's see. So the projects that we have currently are on wheat, tomatoes, zinnias, celtus that we're doing with second generation seeds and Kristen Leach, which I'll talk about briefly in a minute, and spinach. We have a number of varieties that have been released or are going to be released in the coming year. 2 zinnia varieties that have just come out, 2 tomatoes that are in the pipeline, and several wheat varieties that we expect to release either later this year or early next year. And we also collaborate with some other seed projects that are ongoing at the student farm and in seed saving workshops and so on. Scope zinnias. The zinnia project is led by Will Hazard, who was the initiator of the project several years ago when he started his PhD. He's doing his PhD on almonds, so something that's completely different from zinnias, but he moonlights over in the zinnia field. And Cree, who you saw earlier in the alfalfa project, she's also working on zinnias as well. And so we have 2 new releases, UC Shirley and UC Estelita, that we are in the process of commercializing, getting out the door. So hopefully they'll be in seed catalogs for everybody next year, although we'll see how fast we can get things moving. We have other zinnias in the pipeline, and hopefully there'll be a string of zinnia releases over the years. Tomatoes, we have a number of tomatoes that are in the pipeline, and these 2 inbred tomatoes, the UC Orange Creamsicle and UC Rock and Roma, are the ones that we're releasing now. We also have some hybrids that we've been developing. And so these have been screened out in the field. And for Davis and down in CalPoly Pomona, we're looking at tomatoes that can tolerate and produce under high summer temperatures. Over on the coast in Santa Cruz, something that can tolerate the cooler conditions over there. And so the tomato project's been going on now for a number of years, and we have a whole full pipeline of new varieties coming through. Spinach, Danielle, who's also working on a PhD in spinach breeding, is leading the spinach project, largely for bunching spinach, um, and and some specialty things. I'm interested just for fun because I like grit in my spinach. A very Savoyed leaf spinach. I want to see how Savoyed we can get one that's as crinkly as possible. Although I realize it's going to be a mess to get the grit out of it for eating. But anyway, we have a couple populations there that could be released in the next couple years. Celtus stem lettuce is a relatively new project that Mark Watson, a really energetic graduate student, is leading. This is a really cool project because it's a crop that a lot of people don't know about, even though it's very important in many Asian cuisines. And so just educating students and the public about Celtus in itself is fun. And we've been doing a number of trials looking at trying to extend the season when Celtus can be harvested, as well as looking at flavor and so on. And here's some taste tests we were doing earlier this year at the UC Davis kitchen that where we do food. We can all do all kinds of food tastings and things like that. And so looking at different possibilities for Celtus. Laura Roser here in the middle is the student who's really been, or the staff person who started as a student, who's really been keeping Scope going, and she's a key part of the team. And this is Kristen Leach with Second Generation Seeds, a farmer over in Sevastopol who we've been working with, and with her group of farmers to get some of these Celtus breeding materials out on farm to see what people like. Let's see. This is some information about Scope if you want to contact us or check out our website. I direct Scope, but there's a lot of other people who are involved, and it really takes this whole team to make this project work. I think that's all I've got. So if you have any questions, I'd be happy to take them.
Awesome. Thank you so much. I love to check in on what Scope is doing because it's very inspiring and really cool collaborative research going on. I see Tom agrees that Savoy spinach is a much greater flavor. So sounds like you have a market of at least one.
Oh yeah. Well, I think I think some of these are going to be cool. Yeah.
Yeah. If anyone has a question, feel free to post in the chat. I was wondering, could you share a little bit about, I imagine it might vary greatly, but Katharina has, what other qualities are you selecting for with zinnias besides color and petal, maybe specifically in organic systems?
Well, yeah, so a lot of certainly the flashy flowers or particular flower and petal shape and size combinations. Also powdery mildew resistance. A lot of the zinnias are for cut flowers, of course, and so longer stems is always a consideration. I mean, we've talked to lots of flower growers and other people that are interested in, and for some uses like weddings and so on, they want a paler flower color that kind of fits well with the event, rather than things like the old Bennery's Giant, just great big, very colorful flowers. So a lot of the breeding goals are toward maybe muted colors, pastels, that type of thing. But there's, one of the things about Scope is that students can also get involved, and if there's certain things they like, just because they like it, we can go down that road as well. So it isn't constrained necessarily by what everybody wants. We can make up new things too. But those are the big things.
Awesome. Well, thanks so much. Yeah, big round of applause for Charlie. I love hearing about the work that you're doing over at Davis and with the Scope project. Yeah. So we're gonna jump over to Matthew Grieshop as a close for our first Organic Research Updates Flash Talks. He's the director of Gribb Family Center for Organic Production and Research at CalPoly San Luis Obispo.
All right. Can you hear me?
I can hear you.
Okay, fantastic. And can you see some
And
slides?
I can see a slide. No presenter
Right,
mode.
fantastic.
Yep, looks good.
I've got two screens going, so if I'm looking askance, that's why. So what I'm going to talk about today is a project that a recent one of our recent master's graduate students completed as part of her master's research. So the person really responsible for this research is Megan Whittle, who's now working in Santa Maria with the Santa Maria Grower and Packer Association as their scientific advisor. So Megan's project really focused on leaching risk of nitrogen in organic vegetable production, which this is a really big issue in the Central Coast because of water regulations. And so the local water districts have been moving towards a mass balance calculation where growers are basically told you can put this many units of nitrogen on, and it's sort of based on how much nitrogen you harvested out of your crop. And you know, it makes, and the assumption is any nitrogen that's not accounted for in the crop leached into the water, which isn't necessarily a great assumption. And for organic producers in particular, who rely more on you know, natural nitrogen mineralization processes, there's a lot of questions on whether this applies. So I'm just going to present a couple of data from a couple of trials that Megan completed. The first of these really looked at the amount of leaching under organically managed broccoli blocks. And this trial also served as kind of a test for an approach that she used that I'll talk about in a little bit. So the way that Megan assessed nitrogen leaching was with these accumulator devices. And devices sounds really complicated. Really, it's a piece of PVC with some mesh and a proprietary mix of quartz sand and a resin that has a high affinity for ammonia and nitrate. And we actually had to sign an NDA about this method. It's a method that was worked out by some German soil scientists, and so I can't really go into great detail about the methodology. But the short notes is you dig a soil pit, you put these into a profile next to the soil pit, so that you get a column of undisturbed soil above the accumulator. You let them sit for about 6 months, and then you go back and dig them up. So it's a really labor-intensive process, but the cool thing is that you get sort of a quantitative measurement of how much nitrate or ammonia has accumulated in that particular volume of soil over time, because normally it would just continue to run through. With the resins, it captures those ions, and then you can take it out, extract it, and measure it. So the first trial she did was really just a proof of concept for us. We put it under some organic broccoli at our CalPoly organic farm. And the big lesson we got from that was if we used the Ag Order 4.0 sort of calculation like mass balance, this is what we would have calculated in terms of pounds or units of nitrogen per acre. What we actually found with our SIAs, with our accumulators, when we extrapolated up from multiple accumulators under a single field, was that really probably we were leaching maybe about a fifth of the estimated amount. So that's pretty meaningful, because it really suggests that, and this is ground that's been certified organic for about 30 years, receives a lot of compost, has pretty high organic matter for our region. It's a heavy clay soil as well. So this really suggests that maybe those regulations aren't really accurate in terms of what is getting into groundwater. So the next trial, and this was more of a replicated trial, this was done on another block. It followed a sweet baby Brock planting, and we wanted to see that, okay, if during the off season, if we put a cover crop into the system, what does that do to the amount of nitrate and ammonia leaching? So this was a split plot design. There were 3 replicates, and basically these blocks of formerly sweet baby broccoli were split in half, and we either put a overwintering cover crop. We used a canola or a rapeseed cover crop. It's a good winter cover for us in this region. Or we did it blank. And then the other thing that Megan did, and an army of undergrads, is they went in and they either cleared all residual post-harvest, or they didn't clear all the broccoli trash post-harvest. So the split in the split plot is you've got cover crop, no cover crop. The split is removed all the broccoli trash or didn't remove the broccoli trash. Because, you know, one of the other assumptions of Ag Order 4.0 is that all of that, you know, nitrogen that's bound up in the biological tissue, in this case a sweet baby Brock, is going to mineralize and drop down into the water table. So this figure, I mean, this is one of these experiments where it's a tremendous amount of work that compresses into one little figure, which is kind of depressing for the grad students, but makes for a simple story. So this figure shows you on the left our cover crop, where we had that canola cover crop in place all winter, and where we had bare ground. And then the split within there is either where residue was allowed or removed. And so again, if you look at where we're at, we did achieve some higher rates of leaching in the residue bare ground trial. I mean, this is getting to about 25 pounds of nitrogen, which would be about 50% of the estimate by Ag Order 4.0. But where we had a cover crop, the big sort of take home is the cover crop is absorbing most of this free nitrogen, and it's not going down to the 4-foot level where we were sampling with our accumulators. There was a significant main effect in this experiment because we didn't really probably have enough replication. We were not able to show a significant interaction effect where, so basically, the residue removal didn't float out of it. But from a practical standpoint, what this is really suggesting is that, removing residue will reduce nitrate leaching and by quite a bit, but keeping plants in the system is probably a much better fit. And so the importance of this data for us, and we're continuing to think about nitrogen leaching, is really how do we provide policymakers with the data they need to provide organic growers with nutrient management guidelines that don't sort of kneecap them. Because at the moment, there are some discount factors for pelletized fertilizers or composts, but they're honestly probably still way too low. And so the other problem, of course, is if you're trying to build organic matter in your system and you remove all your residue, well, there's really one, unless you're putting a lot of compost and cover crops in, you've just removed one of the major sources of carbon to go into that system and become more recalcitrant organic matter. So the last little bit of data I'll show you, this was actually, Megan took deep soil core depths out of the same treatment, this same experiment. And so you've got your cover crop and your bare ground. She went down to about 3 feet, 3.5 feet, or 100 centimeters, 1 meter. And the take home here is that, again, the cover crop really reduced a lot of leaching down to that level, and it was pretty consistent across the sampled levels where she took a big core and cut it into segments. And on bare ground, you see sort of a spike of mineralized nitrogen in that 10 to 40 centimeter range. And this actually corresponds pretty well with another graduate student study. It was looking at irrigation water and nitrate and role in organic fertility. And we were basically in that study, what we found was that using an evapotranspirative modeled irrigation versus a calendar-based irrigation allowed you to keep nitrate in the rooting zone of the broccoli. But this kind of corresponds to that a little bit. So, the big takeaways from this are that the estimates provided by Ag Order 4.0 are probably, at least in our situation, we're vastly overemphasizing the potential for nitrogen leaching, and that cover crop and residue management can potentially mitigate this, with cover crops probably providing a bigger bang for your buck than residue management. Yeah, and so the center is involved in all kinds of research. We're actually gearing up for our field day, which will be 2 weeks from Thursday on July 24th here at the CalPoly campus. I put a link in the chat if you want to register for that or learn more about that. It'll take you to our main webpage. If you're interested in other research we've done, we put up all our programs from previous research, and every year at that field day, we do a pretty good job of kind of providing an overview of the types of research we've been doing. Yeah, that's it. Hopefully I didn't talk too long.
No, it was great. Yeah, I really love seeing the important work that y'all are doing and the impacts that it has for policy and for organic farmers across the Central Coast and beyond. So thanks so much for sharing. We are at time, so I'm just gonna pass it back to Katharina for a couple closing thoughts. But yeah, let's give a big round of applause to everyone. We really appreciate you taking time to come and share your updates with us.